Good morning. It's February 23rd, and this is your daily brief in science. Here's everything you need to know. A new look at MYC reveals separable roles. Its growth-promoting activity can be uncoupled from an immune evasion mechanism, opening a potential path to target cancer without disabling MYC's normal functions. Researchers show that inhibiting MYC's RNA-binding capacity could expose tumors to immune attack while preserving its transcriptional work in healthy cells. The study, published in Cell on January 22, 2026, led by Martin Eilers with collaborators from JMU, MIT, and Würzburg University Hospital under Cancer Grand Challenges, COUDAC, identifies a distinct RNA-binding region responsible for immune silencing separate from the growth domain. Blocking the immune-evading pathway may offer a more precise cancer strategy than total MYC shutdown, which risks harming normal cells. The RNA-binding region, known as RBI, drives multimerization and RNA degradation without altering transcriptional activation and is central to suppressing innate immune signaling. Under cellular stress, MYC relocates from DNA to nascent RNA, forms multimers around double-stranded RNA and R loops, and recruits the nuclear exosome to degrade problematic RNA, preventing immune activation. In cell culture, both wild-type MYC and an RNA-binding deficient mutant can drive proliferation, but only the wild-type MYC represses innate immune gene sets linked to NF-kappa B and interferon pathways. Future work will determine how immune-activating RNA-DNA hybrids exit the nucleus and how MYC's RNA-binding activity shapes the tumor microenvironment, with clinical applications years away. The research was funded by Cancer Research UK, the Children Cancer Free Foundation, KICA, INCA, and an ERC Advanced Grant. In an orthotopic mouse model, tumors with normal MYC grew rapidly, while those with the RBRII mutant regressed dramatically in immunocompetent mice, underscoring immune recognition as a key driver of tumor regression when MYC's RNA-binding function is disrupted. A newly described iguanodontian dinosaur from China shows hollow, cutaneous spikes embedded in its skin, the first observed hollow spines in a dinosaur. The fossil of Haolongdongi, dating to about 125 million years ago, preserves keratin cores within the spines and comes from northeastern China. Researchers describe Haolongdongi as about 2.5 meters long and herbivorous, with spines along parts of the body that include horn-like projections. The study, published February 6, 2026 in Nature Ecology and Evolution and summarized by the University of Rennes, notes that the spines are not solid bone extensions. The hollow spines may have served multiple roles, including defense against predators, increased surface area for thermoregulation, and possibly sensory perception. The exact relationship between such skin structures and feathers or plumes in dinosaurs remains an open question. Further work is needed to determine whether adult Haolongdongi retained the hollow spikes and to clarify the full functional range of these structures. This discovery expands the known diversity of dinosaur skin coverings and suggests greater architectural variety and integument than previously documented, with implications for understanding dinosaur biology. The findings were led by Jiandong Huang and colleagues and contribute to a broader discussion on how dinosaurs used skin adaptations beyond feathers. As this is the first hollow-spined iguanodontid, researchers emphasize caution in generalizing these traits to related species without additional evidence. A team from the University of Surrey reports a surprising result for sodium-ion batteries. Keeping water inside a layered sodium vanadate hydrate cathode nearly doubles energy storage and enables faster charging compared with traditional dehydrated cathodes. Hydration appears to enhance sodium-ion diffusion within the layered structure, improving charge kinetics and energy density. The hydrated material remains functional in seawater and enables electrochemical desalination, with a graphite electrode removing chloride ions, suggesting seawater batteries could store energy while purifying water. Tests in saltwater show NVOH continuing to operate and contributing to desalination by removing sodium ions from solution while chloride ions are extracted, creating an electrochemical desalination effect. This dual functionality points to future designs where seawater could serve as an electrolyte, advancing simultaneous energy storage and water treatment with potential cost and environmental benefits. Dr. Daniel Commandeur notes the findings could unlock new possibilities for how NVOH is used, potentially delivering safer, cheaper, and more environmentally friendly energy storage solutions. The study envisions applications in large-scale renewable energy storage and electric vehicles, leveraging abundant sodium and seawater to move sodium-ion technology toward practical deployment. If scalable, this approach could advance grid storage and renewable integration due to sodium S abundance and low cost. The hydrated nanostructured sodium vanadate demonstrates superior energy density and cycle stability, maintaining energy retention for over 400 charge cycles and offering faster charging and higher capacity. Retaining water during processing challenges long-held assumptions and suggests embracing hydration chemistry to optimize performance. The discovery positions NVOH among the top sodium-ion cathodes reported and highlights a path toward potential desalination-enabled energy storage with environmental and economic considerations. A shift is underway in Maharashtra as Innovation Council labs expand beyond engineering campuses to 1,132 aided arts, commerce, and science colleges in the first phase. The program moves education from theory toward applied learning, giving undergraduates structured spaces to identify real-world social problems, design solutions, and test workable models with mentorship. Faculty mentors and innovation councils will guide students from idea to prototype and testing, leveraging ecosystems like Idea Labs and existing incubation centers in engineering institutions. The rollout will be phased, starting with aided colleges and later extending support to unaided institutions to foster interdisciplinary collaboration across diverse student backgrounds. The expansion aims to democratize innovation by giving non-engineering students access to structured research infrastructure that was traditionally scarce. This initiative aligns with the national education policy, promoting multidisciplinary problem-solving and experiential learning across humanities, science, and business, rather than limiting innovation to technical fields. Students will tackle community and industry-relevant problems with projects such as reducing school children's mobile use, digital tools for small vendors, and low-cost environmental monitoring like air quality and water testing kits. The broader goal is to transform colleges into problem-solving hubs where learning is grounded in real-world challenges rather than classroom theory alone. Ctenophores, or comb jellies, are ancient planktonic animals whose lineage extends back more than 500 million years and may represent an early branching animal group, a finding that informs how nervous systems and animal origins are understood. Recent surveys along the Caribbean coast of Colombia expanded regional biodiversity records, using non-destructive underwater photography to document 15 species, including 6 not previously recorded in the area. This work avoids specimen collection to protect these fragile gelatinous creatures and creates visual archives for future reference. The study highlights how advances in genomics, high-resolution imaging, and AI-powered analysis are helping overcome sampling challenges and reveal ctenophore diversity, evolution, and potential regenerative and neural features. Feeding relies on colloblasts that release a sticky adhesive to capture prey such as fish eggs, larvae, and plankton, while some species repurpose nematocysts from consumed cnidarians for defense and hunting. Ctenophores move and glow through 8 rows of cilia, ctenes, that diffract light into iridescent colors, with sizes ranging from centimeters to over a meter. Colombian researchers documented 15 species in Colombian waters, with 6 new regional records, using non-invasive photography and field observations. A 2022 Pristine Seas expedition reported the same number of species and 6 first-country records, emphasizing non-invasive methods due to the delicate gelatinous nature. New evidence from Cambrian-era ctenophore fossils suggests ancient nervous and sensory structures, and some species exhibit bioluminescence, expanding understanding of their biology. Globally, ctenophores inhabit surface to abyssal depths, and current work investigates aging, regeneration, and neural evolution in these ancient animals. Ecologically, they are carnivores feeding on plankton and small organisms, helping regulate populations and linking marine food webs. This has been your Daily Brief in Science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.